Okay, Professor E. Prem Kumar Reddy is a director, Icahn School of Medicine at Mount Sinai, New York. Professor E. Prem Kumar Reddy's work at the National Cancer Institute led to seminal discoveries in understanding the molecular basis of cancer, including pinpointing the changes that proto-oncogenes undergo to become cancer-causing viral oncogenes. He also pioneered the development of small molecule inhibitors for cancer therapy, with one drug currently in phase 3 clinical trials and others expected to enter clinical trials soon. His recent work on CDK4 has shown promising results in reducing tumor burden for several metastatic cancers. Dr. Reddy founded the cancer journal Oncogene, as Dr. Suresh said, in 1986 and served as its editor from 1986 to 2009. In 2010, he founded a second cancer journal, Genes and Cancer, for which he currently serves as the editor-in-chief. Sir, I think you are the perfect bridge between industry and academia, and you are walking the talk. So please share some couple of words of wisdom.
Thank you. Thank you very much. I'm really honored to speak to you at this late hour. First of all, I want to thank Dr. Redana and the organizers of this workshop for inviting me. And when I was talking to Dr. Redana, he told me that this is really meant for students and future entrepreneurs who want to start new biotechnology companies. So those of you who are leaders in pharmaceutical industry, please pardon me for some of the simple slides you're going to see. Okay, so let me share my screen, and I hope you can see the screen. And before I start with my talk, I want to introduce you to a series of webinars that are available to anybody and everybody on the website. This is a number of webinars organized by Dr. Sharad Verma, another Indian scientist leading the drug discovery programs at the National Cancer Institute. There are a number of webinars that you can watch on the internet. And I particularly recommend Dr. Caroline Buser-Dobner's talk. She's from GlaxoSmithKline Beecham, and she tells startup biotechnology companies as to what they are looking for in small biotech companies. So this is a very familiar slide for many of you. The drug discovery takes many, many steps, starting from target identification to optimization of the lead and followed by preclinical studies. And actually, there is a law called Moore's Law. This was postulated by Gordon Moore, who is the founder of Intel. He just a few days ago, he passed away. But he predicted in 1965, the number of transistors in a chip are going to double every year. And actually, if you can see, this graph shows that he was fairly accurate in his prediction. And unfortunately, we were expecting a similar breakthrough in drug development, and this has not really happened. It has happened for the initial phases of drug development, which involves preclinical studies. But clinical trials, even today, are very time-consuming. And actually, there is a new law that has been proposed, which is called Irum's law, which is the opposite of Moore's law. If you reverse Moore, it is Irum's law. This actually, this graph shows in 1950, when pharmaceutical companies spent $1 billion, they were able to get anywhere from 50 to 100 drugs approved. Whereas today, typically industrial average is it takes roughly $2 billion to have one drug approved. This is not because of the lack of molecules or lack of technology, but FDA has tightened the rules associated with the toxicity testing and efficacy testing following this disaster that we saw with thalidomide. And since then, over a period of time, FDA has been getting more and more strict. And the second reason for this cost increase is now a large number of molecules are entering clinical trials, and almost 45% of them which enter clinical trials fail in phase 2 because of poor efficacy. And another 55, those that of the 50%, another 55% fail in phase 3 clinical trials. So this has been a major hurdle in pharmaceutical industry. And there is a lot of effort, both from academia and pharmaceutical industry, as to how to overcome this problem. And I think now we know how to make this a shorter trial and a shorter period in terms of drug development because of the advances in technology that we have seen. So when you start thinking about the percent success, the success rate in each step is fairly high. For example, when you identify a target to isolating a hit, there is nearly 80% success and 75% success hit to lead and lead optimization. But as I said, by the time you reach, if you start with 100 molecules, by the time you reach the endpoint, only about 4 molecules are successful in terms of reaching the market. And this has been one of the major hurdles in pharma. And all our attempts are now how to reduce this cost as well as reduce the amount of time required. 13 years is a little too much of a time to spend on developing one drug. So just talking about targets, initially all targets, 100% of the targets until about 1950s were discovered by academia. This is most academic scientists were not interested in drug development, but they were interested in understanding the mechanisms by which the disease process. And in that process, they discovered a lot of what we now know as targets for disease, for development of therapeutics. So here is where the big difference. I mean, of late, the industry has caught up, and roughly about 50% of the targets come from research and industry. But when you look at the performance academia versus pharmaceutical and biotechnology industries in advancing their discoveries to actual discovery of drugs, we as academicians are performing very poorly. So less than 5%, of discoveries are really patented by academia. And even then, when they find a lead molecule which can act as a drug, the number of compounds that actually reach IND stage are extremely limited. Whereas industry, on the other hand, have taken a lead role in this and have been able to really do a very good job of bringing these molecules to the market. So one of the reasons I said there are 50% of these new targets are now coming from industry and biotech. This is because of the advances in genomic technologies. And there have been large number of genetic databases that have been developed by both academicians and industries, and a large number of genome-wide association studies have developed, and which have given us hints about what protein constitutes a very good drug target. And similarly, there have been advances in phenotypic screens and also functional genomic screens. So these are all mainly conducted in industry, and so roughly 50% of the targets are now coming from industry. So this is actually a good example of genome-wide association studies that led to the understanding of the non-alcoholic fatty liver disease. So when a genome-wide association study looking for SNPs, single nucleotide polymorphisms, were performed with roughly about 1,500 cases of the patients and about 17 to 18,000 normal individuals, is one gene was found to be very strongly associated, changes in one gene seem to be very strongly associated with this disease. So when they sequenced this gene, essentially what they found was many of these patients had a mutation. This is a gene that codes for, it's called PNPLA3, patatin-like phospholipase domain containing protein. It's a phospholipase. It metabolizes the lipids. And to validate that this is really a target, people have generated mice, genetically engineered mice, where they introduce the mutation that they see in these patients. And they could show that there's enormous accumulation of lipid droplets in liver compared to a wild-type mouse. So as a result, this has become a major target. This is a drug which is likely to have a market value of $10 to $20 billion. And so, as you can imagine, a large number of pharmaceutical companies are actively developing inhibitors for this particular gene. So this leads to a question: what makes a good target? And now we are slowly realizing if there is a strong genetic association with the disease, this not only makes a good target, but also reduces the time required for drug development and the amount of clinical trials that need to be done for these drugs. So a second important requirement is the target contains a site capable of binding a drug-like molecule or an antibody, especially if it is an extracellular protein like a receptor. A binding of drug-like molecules should result in the inhibition of the biochemical activity of this particular protein. And this inhibition should result in the modulation of the disease process. That means if it's a cancer cell, this drug should be able to block the growth of the cancer cell or induce the death of the cancer cell. So if you look at all the small molecules that have been developed for various diseases, they really fall into a very small number of categories of proteins. As you can see, the GPCRs, G protein associated, receptor associated proteins have been the major targets of many of the drugs that have been developed. These are associated with 7 transmembrane proteins. The next two groups are the ion channels and nuclear receptors. And then a fourth family that is emerging as a very good target is the kinase gene family, especially in the area of oncology, that this has shown really a great value in terms of application. So if you look at the human kinome, we now know there are roughly about 500 genes, and about 100 of them are associated with cancer. So these other 400 genes, the concentration of all the efforts so far in the last 2 decades have been in these kinases associated with cancer. Very little effort has been done to target kinases in other diseases, and this still remains as a low-hanging fruit for developing drugs for various other diseases. But first, basic researchers have to tell us, from academia, what these kinases, various kinases play, which kinases play a role in what type of a disease. So I think there is a lot of work that needs to be done in this area, both in the basic area as well as in drug development. Again, why are these kinases considered to be low-hanging fruit? It's mainly because they are very simple enzymes. They bind a ATP at one end and bind a substrate at another end and transfer the phosphate group from ATP to the substrate protein. So if you can block this particular process, essentially you have blocked the disease that arises from this particular kinase. A very famous example is the BCR-ABL kinase that was discovered in the 80s and which is associated with chronic myelogenous leukemia. And the first targeted therapy that was developed was Gleevec by Novartis, which binds to the ATP binding site and blocks ATP from coming and binding and blocks the entire process. And this particular drug has literally cured CML in most of the Western world. And I will come to countries like India. There are still some problems, but there are easy solutions for these problems. So, we now, as a result of this big discovery of Gleevec, we now have the X-ray crystal structure of roughly about 300 of these kinases. So if somebody wants to develop a kinase inhibitor, a structure-based kinase inhibitor, it has become a fairly simple process in terms of designing compounds that can act as inhibitors. So, there are now a large number of genetic mouse models that have been developed to look at the validity of these targets which are disease relevant. So many years ago, in 2005, one of my graduate students, Harita, who is from Hyderabad, she came to do a PhD with me, and she created actually, one other graduate student, Sushil Rane, created a CDK4 null mutant mice. Essentially, what he did was delete this gene from a mouse and create this CDK4 negative gene. And one of the things we observed was these mice were unable to produce milk when they become pregnant. So it clearly implicated that this gene is severe, plays a major role in the development of mammary glands of the breast. So we Harita mated these mice with a cancer gene called MMTV-NEW. The NEW is the ERB2 gene, which is overexpressed in 30% of breast cancers. And MMTV is a promoter that allows the expression of this gene just in the breast tissue. And when she expressed this MMTV-NEW gene in wild-type mice, which are positive for CDK4, within 2 to 3 weeks, we could see these metastatic breast nodules developing in these mice, which actually after about 3 months, they kill the mice. On the other hand, what she observed was the CDK4 negative mice lacked any of these nodules that were otherwise induced by MMTV-NEW. So based on this, a number of companies started developing CDK4 inhibitors. And there are at least 3 CDK4 inhibitors that are now approved for treatment of breast cancer. And one of our CDK4 inhibitors is currently going through clinical trials. It is in phase 2 phase of the clinical trials. And so this kind of a validation gives a lot of confidence for industry to allow them to make a decision to spend a large amount of money on the development of this particular drug. So, once you have a screen and develop the hits, you really have to qualify the hits. You really have to show, provide evidence that your molecule binds to the target, and it binds selectively without any undesirable mechanisms. That means it should not go and bind non-specifically to multiple proteins in the cell and create a havoc in other cellular metabolic pathways in the cell. So once you have identified hits of sufficient quality and number, you can increase the potency by doing more organic chemistry and more selectivity assays and developing structure functional relationships, you can develop a lead molecule. So this has become a fairly rapid process today because of the large number of compound libraries that are available, crystal structure of the targets that have become available, especially for kinases, and the ease with which you can manipulate, do chemistry in terms of increasing the potency and selectivity. And for other diseases, for other protein targets for which we really don't have a clue as to how to make a compound, an inhibitor, a number of other compounds, other technology have come up. One of the very popular technologies is the affinity selection mass spectroscopy technology, where you mix your compound with a recombinant protein. Nowadays, it's very easy to make recombinant proteins. And then you subject it to size exclusion chromatography, identify, isolate the target protein and look at the compound that binds to it. This being routinely used by pharmaceutical industry who have compound libraries of millions of compounds, where you cannot afford to screen each compound against a particular target. Similarly, a DNA-encoded library technology is becoming very popular. Here, you can attach a barcoded DNA to your molecule and then do the affinity matrix pull down of these molecules that bind to your affinity and identify the molecule that binds to it by PCR, and then do further evaluation after large-scale synthesis of these compounds. These technologies, which are relatively new, have become very popular now and are being extensively used, which is likely to create a lot of hits for so far unreachable targets. So phenotypic activity-based screens are very popular in cancer. So like I told you, there are only 100 kinases that seem to play a critical role in cancer. And large number of groups have developed compound libraries which inhibit these 100 kinases. For example, in our lab, our chemists have created a library of roughly about 5,000 compounds. And these 5,000 compounds, I'm pretty confident they inhibit virtually we have candidate inhibitors for all the 500 kinases. But how do you select them? So here, what we do is let us say you are trying to find a drug for AML. You plate the AML cells in, a bunch of AML cells, 3 or 4 different cell lines in these plates, and followed by normal cells as well as cells of other types of cancer, and screen your compounds this is a medium throughput screen, roughly 96 well plates, and you can perform this within a week and identify all the compounds that can inhibit the growth of a particular cell line you are looking for. Here you are looking for a compound that very specifically kills AML cells, but none of the normal cells or other cancer cells. And then once you identify, you can send it now. Fortunately, you have CROs who have set up these kinase assays for all the 500 kinases. They will run through their screen, and within a week, will let you know what kinases your compound inhibits. You can take these kinases, do basic studies like crystal structure or optimization using organic chemistry, and then do mechanism studies followed by animal studies. So I just want to illustrate to the students one example of this kind of a technology. So myeloid leukemia, we were very interested in myeloid leukemia because there is a very defined genetic marker for this. There are 2 kinds of myeloid leukemias: chronic myeloid leukemias 100% of these patients have mutations in the BCR-ABL gene. Dr. Redden has done a lot of work on the BCR-ABL gene. And in the case of AML, 30% of AML patients have mutations in the FLT3 gene. So I told you, Novartis developed this Gleevec, which is inhibitor for this BCR-ABL gene. It literally cured this disease in Western countries like United States and Europe. But there seems to be a very large percentage of Indian patients who are resistant to Gleevec. This is because of acquisition of mutations in other parts of the BCR-ABL. And so, Novartis and as well as other companies like Bristol Myers Squibb have developed second and third generation BCR-ABL inhibitors. But unfortunately, these are still under patent, and none of these drugs are currently available in India, at least in an affordable manner. And there is, for whatever reason, as a function of time, there has been an increasing incidence of CML and AML in India. So lack of availability of drugs for Gleevec-resistant CML. Gleevec, actually a generic form of Gleevec, is produced in Hyderabad by NATCO, which is actually doing extremely well in India. But the lack of second generation drugs for Gleevec-resistant CML and FLT3 positive AML in India makes it a very important medical need. So we thought we will try, because we have this large library, see whether we can come up with a candidate molecule. And when we did these phenotypic screens as well as enzyme screens, we found several molecules that inhibit FLT3 and BCR-ABL. And one molecule we chose to work with because this had some very unique property. It not only inhibited FLT3, but also inhibited ABL, BCR-ABL, and SRC kinases. All these kinases play a role in leukemias, and we thought, we will advance this compound. And interestingly, what we also found was this compound inhibits all mutants of BCR-ABL. We are still trying to get a crystal structure to try and understand why this particular compound is so effective with all these mutants of BCR-ABL, and we will know pretty soon once we have the crystal structure. So, and when we did these screens with other cell lines, there was a remarkable specificity for this particular compound. So this is an AML with a FLT3 mutation. It killed these cells at a GI50 of 10 nanomolar. And this is a BCR-ABL positive cell line, K562. It killed this cell line with a GI50 of 40 nanomolar. On the other hand, these are all AML cells, but with these three cell lines are AML cells, but they have wild-type FLT3, so this compound had no effect really on the viability of these cells. Similarly, it had really little or no effect on any of the other cancer cells. And again, I'm not showing the data, but none of the normal cells were affected by this compound. So this remarkable specificity is a very good strength, because this is, it is already telling you this is unlikely to have major side effects associated with chemotherapy. So this is typically what we do in terms of efficacy studies. You inject tumor cells into these mice, called nude mice, which don't have hair. That's why they're called nude mice. And once the tumor develops to 100 to 200 millimeters size, we start treating either with placebo or with your drug and start measuring the tumor size. This is a cell line which is very aggressive in growth. As you can see, in placebo-treated mice, it grows enormously. So when we administer our drug orally, this is orally bioavailable, what we were very surprised, we rarely see this with the other targeted therapies, including Gleevec. within 4 days, the tumor had completely disappeared. So this was somewhat, I was very skeptical when my postdoc came and told me that this compound is able to eliminate tumor growth. Most of the often, these therapies don't eliminate tumor growth, but reduce tumor growth. So, we went to see the mice, and certainly what she was telling was true. And then, we had these other mice, placebo mice, where the tumors had grown to an enormous size, roughly 1,000 millimeters. So compared to this, if you look at the placebo mice, this is the size of the tumor. So we decided, why not we take these tumors and administer this drug to these animals, which we had to sacrifice otherwise because animal IACUC rules do not allow us to keep these mice with large tumors alive because it's considered to be inhumane. And again, what you can see is we could see by day 4, it was considerably reduced. And by day 9, the tumors were gone. By day 10, by day 9. This is the progression of the tumors. Not only didn't it go away, but when after about 21 days, we start treatment, and we did not see for the next 2 months reappearance of these tumors. So my postdoc was extremely happy, and she was telling me, oh, we got a drug for AML and CML. So unfortunately, I had to remind her, this is the start of the journey, not the final destination of the journey. So here now, once you have identified a lead, then you have to answer several questions. That means more work needs to be done. So we have to show the compound binds to the target with the desired selectivity, which we could show because these are kinases. We took 40 other different kinases and showed this very specifically binds to only 3 or 4 kinases. And the second question we have to show is, does the compound reach the desired organ? So a lot of times when you are dealing with diseases like pancreatic cancer, people develop drugs that can kill pancreatic cancer cells in animals and in vitro. But when you actually do clinical trials, what they find is this drug never reaches pancreas. So it has no effect on the pancreatic cancer of the humans, because here in animals, we are injecting tumor cells subcutaneously, which is not really pancreas. So these are the questions that we need to answer, which is a time-consuming exercise. We have to show how long the drug stays in the system, how long it stays in the blood, how long it stays in the organ, and that the compound exhibit efficacy in relevant disease models. This is where our genetically modified mouse models, like what I have shown for breast cancer, become very useful. More recently, these PDX models have become very popular. These are patient-derived xenografts where tumor cells are taken out of the patient and directly injected into the mice, immunocompromised mice. And people have shown they form tumors, and you can treat these tumors very often. For example, in PDX models, what people do is take the tumors and directly inject. For example, if it's a breast cancer, they inject it into the breast so that it is organ relevant. And when you are testing a drug, you also are testing whether the drug reaches the desired organ or not. And then we have to go to what is the best formulation that can be used to deliver the drug. India is an expert in developing formulations, and I'm sure there are a lot of CROs who specialize in this particular field. And more importantly, finally, you have to answer the question, can genetic data be used to identify subpopulations of patients? This is the reason why we chose FLT3 mutant CMLs, because I can simply go to a group of AML patients that are positive for FLT3 and test this drug on those patients. The first 20 patients will tell us whether the drug is going to work or not. A yes or no answer you are getting right in phase 1 trials. So this enormously reduces both the cost and time required to development. So now at least in cancer field, the emphasis is now looking at drugs that can be used for treating subpopulations of patients that have a very distinctive genetic marker, so that you are not wasting time testing on patients who are not going to respond to the drug. So, once you have done these, to satisfy FDA, you have to do what is called IND enabling studies. So FDA requires you to manufacture kilograms of this drug, which is more than 98% pure. So you have to provide proof that you are able to make kilogram quantities of this drug, which is quite often, in most cases, is 99% or more pure. And then you have to determine what is the maximum tolerated dose in rats. And you have to take 2 vertebrate animals. In oncology, most of these times, what we do is in rats and dogs, or rats and monkeys. So here you have to repeatedly dose for 7 days in the rats and look at toxicity associated with the administration. Once you pass this test, you have to do a 28-day toxicity and toxicokinetics. That means how much of the drug is reaching the blood, how much of the drug is reaching each organs, that kind of a study. These studies normally cannot be done in an academic lab. It becomes very expensive and very tedious. So there are a large number of CROs, both in India, China, and United States, who do this for you in rats and dogs. And the cost approximately is anywhere from $700,000 to $1 million. So probably in India, it's about a third of this cost, or 1/2 of this cost. And you have to be prepared to spend this kind of money. Now you have identified a very strong candidate. So the preclinical evaluation, which is done both in the lab as well as by CROs, is you have to come to a conclusion, determination of the most efficacious dose, show efficacy in xenograft and PDX models, and if possible, in genetically modified mouse models. And this will allow you to submit an application to the FDA for approval for phase 1 clinical trials. So, the therapeutic index is a phrase that you are going to hear very frequently in drug development industry. This is the difference between the most efficacious dose and the most maximum safe dose. So that's why, you have to show a therapeutic window. That means your drug should be able to treat the disease without causing too much of toxicity or side effects in the patients. And I think it is this is where majority of the drugs today fail. That's why if we have a very genetically identified marker and a target which is very, very specific for that particular disease, this therapeutic window is likely to be very high, and the chances of success is going to be also very high. So, here is where, it is becoming very clear. The academia have certain strengths and pharma have certain strengths. And, and it has become imperative, at least in the Western world, to develop a new drug. You need collaboration between these two groups. Academia have really very deep knowledge of biology, signaling pathways, and as I told you, targets. And quite often, they're very good in creating the initial molecule inhibitors that work as tool molecules for whether to see whether inhibition of a particular target can achieve an end result. And it is academia who developed these genetically modified mice, xenografts, as well as these PDX models. And also it is now, at least in the United States, it is the academic hospitals that do all clinical trials. The pharma, on the other hand, has wonderful knowledge of medicinal chemistry. This is the storehouse for compound libraries. Many of the companies like Novartis, Bristol Myers Squibb, has compound libraries which run into hundreds of millions of compounds. And more importantly, they have the manufacturing and formulation capabilities and pharmacology, and also financial resources for supporting clinical trials. So it is the combination of these two now that is working most effectively in terms of drug development. So if there is an academician who is interested in drug development, I think as soon as he finds a target, he or she should start talking to all pharmaceutical companies that he or she can approach, and that will tremendously accelerate the progress of research in this field. So again, this is what businessmen wants to see. The pharma cost, it has been estimated by Deloitte, and this is not my numbers. This is some accounting company that estimated these costs. Screening of hit to lead cost them about $1.5 million. And lead optimization, this is an expensive process, costs about $13 million. And very often, pharma want to work with more than one compound. So the preclinical IND development costs about $8.8 million and roughly about $22 million per compound by the time they enter phase 1 trials. Whereas academic cost in these company estimates as zero here, because a lot of this work is done by academia through grants and through their routine basic research that they are doing through graduate students and postdocs. And it's only, they spend money only in this preclinical IND development, which is roughly about $2 million. And so by the time they reach phase 1, there is a tenfold difference between pharma and an academic institution developing a drug. That's one of the reasons why in the United States, most big academic institutions have set up now facilities to raise money to support some of their molecules that are discovered internally so that they can go at least up to IND studies and then start collaborating with pharma. So in the case of pharma, risk-adjusted cost per stage goes up because you are dealing with multiple compounds for the same target. So at the end, they end up spending about $67 to $70 million per target, whereas academic institutions have this cost considerably reduced. And I'll skip this. So it has become very important for academicians to collaborate with pharma if they really want to have their target validated and a drug developed for their favorite disease. So pharma is looking for a right target, a strong link between target and disease. There's nothing like a genetic evidence like what I have shown. Differential efficacy, that means when you take a normal gene versus mutant gene, your compound should be able to not affect the normal gene, but affect mainly the mutant gene. And have available predictive biomarkers for you to monitor the progression of the disease. And it also has to be right tissue. So that means when you develop a drug, you have to make sure this drug gets into the tissue of your interest. For example, pancreatic cancer. the drug has to get into pancreas. And you have to have defined pharmacodynamic biomarkers and a clear understanding of preclinical and clinical pharmacokinetics, understanding of drug-drug interactions, especially if you are going to do combination studies, and right safety profile. So this is done mostly in animals, and it's fairly routine nowadays to do, and the right patient. So this is where there's an enormous economy in cost, because if you have a right patient with a genetically identified marker, the cost of clinical trials dramatically drops down from hundreds of millions of dollars to tens of millions of dollars. And of course, pharma always looks for good commercial potential so that they can make a lot of money. But for academicians, this is less important. And so, I'm also asked to tell a little about what is needed to start a biotech company, especially in United States. You need a strong patent covering both USA, Europe, and if possible, Japan, Korea, China, and India. These markets have, China and India are becoming increasingly important. 10 years ago, most pharmaceutical companies never filed patents in China and India. You need seed funding of $2 to $8 million in United States, probably is much less in India. And most importantly, you need a strong, highly motivated scientific team and strong group of scientific advisors and board of directors with experience in pharmaceutical industry and a clearly laid out clinical path. If you have all these ingredients, the chances of success are very high. So I want to end with a very positive slide. Here is the very first drug, targeted therapy that I told you was developed, was the Gleevec. So this development of Gleevec took 41 years. Dr. Peter Noel in Philadelphia discovered Philadelphia chromosome in 1960, which is associated with chronic myelogenous leukemia. So we cloned and sequenced the BCR, the ABL gene from the Abelson leukemia virus in 1973. We published this in PNAS. And in between 93 and 95, within, the BCR-ABL inhibitors were isolated. And then but the actual clinical trial, there was a lot of skepticism in the drug development, drug pharma companies. And it took another 40 years for them to start clinical trials. And within about 2 years, they could finish clinical trials because all they had to do is identify all Philadelphia chromosome positive patients and do a clinical trial. So however, ERB2 amplification was discovered by Stu Aronson in 1985 at NIH. And within about 10 years, you had a drug and it went into clinical trials. And this took 13 years. Similarly, PARP inhibition, it took another 15 years. But BRAF was identified as a mutation in melanomas in 2002. Within 8 years, they had a drug that was approved by the FDA. And ALK, a mutation which is seen in lung cancer patients, was identified in 2007. By 2010, they had the drug in the market. And I don't have to tell you about the COVID-19 drugs. It took them only 1 year to develop, do a clinical trial, and market it. So I think we are learning how to do this fast and also do this economically, and what are the ingredients required for it. And a lot of these technologies, fortunately, have come from basic research in academia. And I think I will stop there and answer any questions.
Excellent presentation, sir.
Thank you.
This is Raghu.
Hi, Raghu.
Really, you covered a broad spectrum of drug discovery. And particularly like on the kinases, how are you overcoming the selectivity? Because all the, even the 100 kinases of cancer, they are structurally very similar, particularly they are all the same ATP binding pocket. Even Gleevec binds to both ABL and c-KIT kinase. So how are you bringing the selectivity towards a particular kinase?
So, I mean, now the concept is if you can limit it to 3 or 4 kinases, by experience, we know the toxicity profile does not seriously affect the development of the drug. So it's only when it starts inhibiting hundreds of kinases you see problems.
Okay, my other question is, I don't know, like whether cancer was existing even probably 50 years back, 100 years back. Nowadays, we hear lots and lots of cancer. Like it is like the word we hear, like we see like 1/3 is diabetic in India, like hypertension. And we are also hearing the same way, so many cancer patients. Is it because of the change in the food culture or like genotypic? Like what are the things happening? Why this is more?
Our environment, we're breathing a lot more polluted air, and which is full of chemical carcinogens. And also, food additives, there are a lot of additives that are added to food. You may know the name of the term Ames test. Ames was a microbiologist. He was looking at the dyes that are applied to fruits in United States when they were selling apples. They wanted to make them look red, bright red, so they were applying a dye. It turned out this dye was full of carcinogens. And so all those people who ate those apples, got breast cancer. That was the origin of Ames test.
Sir, I just had one question. Like, nowadays, because of high and technology, a lot of people talk about, antibodies and cell therapy and gene therapy. So
yes,
how do you position small molecules within all these advanced therapies which are actually coming through?
So antibody therapy is very effective, especially if it is, for example, ERB2 amplification. There is a drug called Herceptin, which works quite well. Okay, it's an antibody to ERB2 receptor. It goes, binds, and blocks the cell proliferation. But, antibody therapy is not oral therapy. So you have to go to a hospital, get infusion of the antibody. But now new technology has come where you can link this antibody to chemotherapy and direct your drug to a particular cancer cell, which is overexpressing that particular receptor. And there is a new drug called Enhertu, which is a combination of ERB2 antibody linked to a topo inhibitor, etoposide, and it works wonders for ERB2 positive breast cancers. So as the technology improves, the treatment also is going to be more and more effective.
Hello, Professor. A nice presentation.
Thank you.
I am Dr. K. Nagaya from IIT Hyderabad. So this in big companies of US or UK, they are sponsoring joint research for the schemes for the premier institutes and universities. So it is involving from the professors and the life sciences specialization in drug discovery studies, like virtual molecular modeling or DNA, RNA, enzyme, all these things. So this infrastructure and experts ecosystem existing in India. That is my question. Any industries doing from this type of infrastructure, this type of specialization, and this type of the academic support is there in Indian companies or Indian universities? They are doing this area.
See, I think, the pharmaceutical companies in India right now, as one of the speakers mentioned before, are mostly into producing generic drugs because of the cost involved in developing new drugs. But as India becomes wealthy and these pharmaceutical companies are reaching valuation sizes of $10 to $100 billion, when they have more funds, I think they will venture into new, developing new drugs simply because they're a lot more profitable. And that culture will come with time. And I think it's just beginning in India. And, in one of the things that happened 10 years ago in the United States is companies like Novartis went and signed up research agreements with the major academic institutions. They went and signed an agreement with Harvard Medical School. We'll give you $200 million a year. You can use this for drug development internally, but any patents that come out of it, we have the first right of refusal. So similarly, our Mount Sinai has an agreement with Celgene for $100 million a year for drug development. So I think those type of collaborations will eventually come when there is funds that are dispensable for major pharma.
Great, sir. Thank you so much.
Thank you. Thank you all for your patience.
Yeah. Sure, Dr. Reddy. I think you condensed 30 days, 3 weeks of workshop into one crystal clear, elegant presentation. Amazing clarity of thought. And really, even though I've been in the business for 30 years, thoroughly enjoyed it. And so did all the audience, I think. So really kudos to you for that. Very well done.
Thank you. You're very kind.
And you should get to bed now. Bother you anymore. We're going to move on to the next panel discussion. So have a good night, and really appreciate having you as part of this function. So thank you so much. Yeah.
Professor Reddy, I have a question. I posted it in the chat. This is Sharath from Hans India, a reporter.
Yes.
You spoke really well about the collaboration between the industry and academia. So I just want to understand how startups can be benefited by collaborating with
okay.
academy.
Let me give you my example. I started a company, for example, this drug was developed, this AML drug was developed at Mount Sinai. And there are many advantages for a startup company having a strong collaborative ties with an academic institution. Number one, patents. So we filed a patent in the United States, USA, China, India, Japan. And, if I were to do this as a pharmaceutical company, the fee for filing the patent in US is $10,000, in Europe it is $100,000 for a company. But for an academic institution, it is $1,000 and $10,000. It's tenfold less. Right there, you save several $100,000. And two, what we did was we raised some money in the company. The patent was licensed from Mount Sinai, but we signed a research agreement between Mount Sinai and the new company, so that all the work can be done in Mount Sinai, because Mount Sinai also is interested in getting their patent into the market. That's when they get all the royalties. So, we did not have to build a building and buy all the infrastructure, and all the preclinical studies could be done in an academic environment. And I think Hyderabad University has a very good incubation center. That's a good example of how you can combine both.
Thank you, sir. I think we have to move on.
Thank you.
Good night to you.